So in this next recording, we're going to continue our study of biomacromolecules, specifically proteins, but we want to focus on protein structure. Remember, we've got 4 biomacromolecules: carbohydrates, lipids, proteins, and nucleic acids. We've already talked about the basics of proteins. They're made of amino acids, and that we link those amino acids together in a peptide bond. Now, when we look at a protein, though, we can see 4 levels of structure. And this linkage of amino acids in a linear fashion is actually the primary structure of a protein, or its first level of structure. So it's a unique sequence of amino acids. And the idea of that sequence of amino acids is very important because that's going to be basically the information that DNA encodes for. All right? So again, protein structure is very important to understand because proteins function in the cell are going to regulate the biochemistry of the cell. So again, proteins do stuff. Now that primary structure can be built upon, and we get secondary structure, which is going to be also known as microstructure: alpha helices, beta sheets, and random coil. And secondary structure then can fold in to make tertiary structure, which is the overall 3-dimensional shape of the protein. Several individual protein chains or polypeptides can come together to form what's called quaternary structure, but not every protein has that. And so we can see those 4 levels of structure depicted here: primary, secondary, tertiary, and then quaternary. Basically, that's saying first, second, third, and fourth level of structure. Let's take a look at each of those levels in turn. So again, primary structure, or the first level, is simply the sequence of amino acids. It is inherent to the genetic information. You can give me a sequence of DNA, and I can tell you what amino acids it encodes for, and you'll be able to do that before the quarter is over. So if we look at that, here we have a series of 25 amino acids: glycine, proline, threonine, glycine, glutamine, serine, lysine, and so on. And that's it. It's like beads on a string. Now, what we want to remember is that proteins can vary in size. They're going to vary in the sequence of those amino acids. Some are going to have very few amino acids that are the same, right? So you'd have strings of many different amino acids. Some proteins are made up of just a few amino acids, but lots and lots of them. So there's pretty much infinite variation, even on the primary level. Now that primary level, though, is going to be subject to the chemistry that is in the world, right? So again, the laws of physics are going to govern biology, and this is where we're going to see a lot of that come into play. And all of this is going to drive ultimately the shape of the protein, and the shape of the protein will dictate its function. And its function can be varied, but the key roles of many of these proteins are directing the chemistry of the cell. Right? So primary structure, secondary structure is going to be the result of hydrogen bonding between backbone constituents. Right? So basically we see 2 types of secondary structure that are regular, those being alpha helices and beta pleated sheets. Now those other parts of the protein that don't fall into one of those two categories we tend to call random coil. There are subtleties to this that we will explore later, but for now we're going to keep it in this kind of simplified format. So secondary structure, again, we see that dotted line is hydrogen bonding, linking that beads on a string type structure into a coil or into a sheet. And this then can in turn pack into a tertiary structure. And this is where really the chemistry that we've been talking about as far as hydrophobic, hydrophilic, opposite charges drawing towards each other, all of these things that we talked about, again, as basic chemistry, are going to help drive this protein strands, these amino acid strands, into a glob that has a specific shape. So tertiary structure is that 3-dimensional shape, and we're going to see interaction between the R groups: hydrogen bonding, ionic bonds, these are what we call salt bridges, hydrophobic interactions, van der Waals forces, and then disulfide bridges, which are actually going to be covalent bonds between parts of the protein. So again, when you see these proteins depicted as these kind of blobs, what really you've done is taken those beads on a string and letting them all fall together and interact with each other, right? Interacting how? By the laws of physics. So these are kind of the interactions that you want to remember that are going to contribute to tertiary structure. Again, hydrogen bonding, van der Waals forces, hydrophobic interactions, disulfide bridges, ionic bonds. All right? So that's really, really important. This is a great slide to kind of commit to memory. And as we talk then about the function of proteins, we're going to see how these forces can be perturbed, and hence that affects the function of the protein. Now, our final level of protein structure is quaternary structure, and not all proteins will have quaternary structure. Some will be a single polypeptide chain that folds on itself and is now functional. Some, though, will get together with other polypeptide chains and form a quaternary structure, or a fourth level of structure. Hemoglobin is kind of the classic example where you have 4 subunits, each binding a heme that is then responsible for binding oxygen. And this is a classic biochemistry. You talk about how that molecule works to bind oxygen and release it in tissues that need it, but you'll get that when you take biochemistry. So again, you want to remember the 4 levels of protein structure: primary, which is the amino acid sequence; secondary, which is microstructure, alpha helices, beta sheets, random coil; tertiary structure is the 3-dimensional shape; and finally quaternary structure, which is with only some proteins, when multiple polypeptide chains get together and to form one larger complex.